Hello there. Do we know regulated code, or we are ready to design new life? These questions we'll try to answer today. My name is Alexander Kell, and you are back to Coffee Break with TRANSFAC. And take your cup of coffee and relax. Today we have a very interesting topic. Let me share my screen. Intelligent design of promoters. All right, so I gave this sentence to ChatGPT, and it gave me this nice picture. And actually, it tells me something and reminds me what Yuval Harari said in his bestseller book, Sapiens, that in the future, humans might start playing a role similar to that of the god by designing life. And maybe in several thousand years, it will be difficult to differentiate actually the real life forms and those which are made by intelligent design. The problem, though, is probably it will be artificial intelligent design. And that's a problem. Anyway, do we know regulatory code and why do we need it? Let's talk about this a little bit. So if we have a gene, it's like, gene in a empty space because we know a bit about the protein structure which is encoded by those genes and what is the capabilities of those proteins, but it's very difficult to predict where these genes should be expressed, in which organs, tissue, stage of development, and in which conditions, in which cell cycle phase. It's like if you have a text in some language which you don't know the code, you don't know the vocabulary, you don't know the alphabet, you only know very little pieces of that text and understand them, like names of those elements. But the message, what to do, what this person should do, like me, is encoded in some bizarre language which we don't know. So, but actually, we know probably something, and we know that genes are, expressed and regulated by complex machinery, which is built by transcription factors, and those factors are binding to the promoter areas and to the enhancer areas, which are communicating to each other, and we'll talk about that today. So actually something is known, and what we definitely know is that the core element of the gene regulation are those transcription factors and their binding sites. So they have to be there in the nucleus, these are proteins in the nucleus, like this protein called RNX, and this will be the example we'll talk today. It binds to a particular place in DNA and gives a message to the gene nearby to start or to stop transcription. What we know about those genes, about those transcription factors. So just to remind you, we were talking about that many times. So we know the motifs, actually. In DNA, there is not just a random nucleotide composition where those transcription factors are binding, but actually there are very specific places called motifs. And for instance, if we talk about this RNX transcription factor, and another synonym of that is AML1, it has a very little message, very little motif like this, just 3 times T and 3 times A in between, and something a little bit nearby. And many of those motifs we actually collect in TRANSFAC database, and you can find them also in other databases like in Jasper, Hokomoko, and many others. TRANSFAC, we are trying to collect, considerably from many different resources. But why do we need that? Basically, when these motifs are discovered, first known sites are collected, aligned, and those motifs are reconstructed. And most important now, when we have this library of motifs, and we have in TRANSFAC more than 10,000 of those motifs, the various tools can apply those motifs to find the potential binding sites of those transcription factors in the genes. All right. So why do we need that? And what has this to do with artificial promoter design? By the way, when I start, digging ChatGPT a little bit more, saying, okay, tell me something more concrete about artificial promoter design, it started giving me some kind of such strange pictures where you have some organs, some 3D printer and things like that. So basically, let's actually look around and see what are the areas of application where these artificial promoters are important. So I listed them like this, and you can see this is a list. And by the way, let me first read it, and then I want a bit of feedback from you, and maybe you can tell me what kind of areas are most interesting for you. And these are the areas of application like tissue engineering and organ engineering. It's actually most intriguing when we want to build new tissues and organs like liver, heart, lungs. And of course, artificial promoters are needed there to precise control of the genes, which has to be, working in particular cell types to differentiate those cells in different types of cells. And then a particular organ is a composition of those different types of cells. So similar, but a little bit different is regenerative medicine, where actually we don't substitute the whole organ, but we try to fix the wrong organ or the organ with a disease inside the body. So basically, again, the constructs with a particular promoters or enhancers, they have to be activated. So in this case, special constructs are used, and then the living organisms are infected by those constructs, and then these constructs are ending up in a particular organ or tissue, and there they have to express the protein or a product which they have to express. They have to drive the expression of the gene in that particular subtype of the cell, which is, we want to target. So that's is used in regenerative medicine, and I will give some couple of examples of that. Very important and very interesting, actually, direction is cancer therapy. So where actually you don't need to fix the cell, but rather you have to kill the cell. So the cancer cells are growing, and we want to kill those cells. And the idea is then with artificial constructs, where we have artificial promoters, which again they're designed to start the genes and to give the gene kick off only if they appear to be in these cancer cells, and then these genes are these cassettes. They are kind of designed in such a way they have sort of suicidal product, and then those cells are dying. So it's the idea of using in cancer therapy. Generally saying gene therapy, not just in cancer therapy. So again, maybe the targets could be sort of other types of tissues and cells. It's also similar to regenerative medicine. Yeah, of course, the big area of application is pharmaceutical production. So not only pharmaceutical, but in general biotechnology. So you want to grow a certain plants better. You have to produce different products. So for that, you have to express a given product in a given cell types. And again, you need artificial promoters. And generally speaking, we have this synthetic biology, movement when basically all kind of crazy constructs are created, and basically really artificial life is created, and there are a number of interesting examples of that. And generally, of course, these artificial promoters are used for biomedical research in general, where you want to express something in a given cell type, and you need a vector which will be expressed there. So we want to roll out a pool, and I would like to ask you to answer what is your most interesting, particular interest in which field. Just for curiosity, just put only two, not more than two, and then we'll see a kind of distribution of your interest. to which areas of application of artificial promoters you are most interested in. So here's your pool. So just take your time when it and when it is ready, we'll show the results. But in the meantime, I will proceed. And by the way, if you have any questions in between, very urgent questions, for instance, or you just put your question and I will have time at the end to answer those questions. Please put them in Q&A. Here's a Q&A section. Please find it in your interface, and just don't hesitate. Put any questions of your interest, maybe not even related to this topic, but related generally to TRANSFAC and to our products. Just tell us what is your interest. All right. Okay, so let's go further. And so let's go just, bit a bit towards those application areas. And I want to give you a couple of examples. And by the way, today is a little bit different from usual coffee breaks. I have a lot of material here, and I decided to give more like a lecture at the beginning. And maybe at the end, we'll have some time for online demo and also, of course, answering your questions. And of course, if you're watching this video on YouTube, just give your like, so it's very useful for us. Anyway, so if you talk about regenerative medicine and synthetic biology in general, so I was reading a lot of papers and review articles, and I grabbed some interesting pictures like this, which shows you basically the dynamic. It's very recent development, which is happening in very few recent years, like booming of this regenerative medicine and general synthetic medicine. So you see here, it was started, just with IPS, embryonic stem cells, and using embryonic stem cells initially to transform them to other cell types of cells. Then, of course, this very big breakthrough of induced pluripotent stem cells when we found a way how to, take a normal cell, standard, not embryonic stem cell, but the standard cell, and differentiate it. And those cells became pluripotent. So basically, they have all potential to be developed into various different types of cells. And already from that time point, there was a very interesting ideas and very big question, how to trigger those cells to differentiate a particular type of cells. And people did a lot of experiments doing like blindly different kind of conditions. And they found many components how to kick the cell in different line of development. In most cases, this was done without understanding what we are doing. It's just randomly selecting conditions, different compositions of actually transcription factors. Finally, they found 4 factors which are most important, and so forth. But today we see like a booming area when we are, it's possible to generate organoids of different complexity. And sometimes it's amazing when just starting from a single cell, people start creating almost already organs which are moving, which are, beating like heart. And here we are talking about, organ on a chip, when the different type of cells are composed together in a kind of a machine or kind of a device, or even a human on a chip. The whole different type of cells from different types of organs are put in one device, and they communicate to each other. So I don't know if pool is ready. Maybe we can see the results. So let's have a look. All right. So we see here. I hope you see it, right? If you don't see, just give in the chat like minus, or tell me you don't see it. But if you see it, it's very interesting to see that a lot of you are interested in cancer therapy, 61%, gene therapy, 72%, right? And tissue engineering and regenerative medicine is a little bit less, but it's also part of your interest. Very good. Thanks a lot. Thanks a lot. And that's let's proceed. Okay. So what is actually what are the tools of those synthetic medicine and synthetic biology? It's very simple usually. So just triangle or triple, I would say. There is a cell, and there's a different type of receptors on those cells, and we know how to trigger those receptors with some activators, with maybe even with a light, which I will show in a moment. And then those receptors will trigger transcription factors, make them active. And then those factors would bind to a gene of our interest or a set of genes of our interest with a promoter, which is designed in a specific way that these transcription factors will bind there. And there are two ways of doing that. Either we use endogenous transcription factors, so those factors which are expressed in those types of cells by itself, or very interesting actually to do it synthetic TFs, or TFs taken from another organism. So basically, which were never appeared in this cell types or even not appeared in these organisms. Why not using them here? And then they will bind to very specific targeted cell gene and promoter, which is not bound by any other transcription factor. That's very interesting, way of doing it. And I would like to show one example from my experience. We had a project called optogenerative, so where we were involved in designing the implant, some kind of chamber, which has to be, implanted on the skin in the future in human, but of course we did it in mouse. And this chamber have special designed cells. And this chamber has actually an antenna. So you can give a signal from your mobile phone, more or less, to this chamber, which is under your skin. And this chamber will start lighting, give a light, like red light on the cells. And those cells are specially engineered. So they have a receptor which accepts those red light. And then there was our part of the story. We have to design a special promoter which would accept or optimize those promoters which they have already, which would accept that signal through this signal cascade. And we were modeling that system. So that was a very, very interesting experience of, involving in the real, a real design of a new life sort of thing. Okay, but there are more bizarre things which people are doing with this synthetic biology. So when those constructs, when one cell is actually expressing particular receptor and another cell is accepting this signal and then transferring this signal to activate a special set of transcription factors, which are in turn activating genes expressing another receptor, which will be targeting another type of cells. So you can create so many different types of cells and then put them all together, and they will start self-organizing. So they will find each other, and they will be sort of organoid, which have a structure. So certain cells will be inside, and other cells will be outside. So this is a very, very interesting to observe how this life is actually created from your design. Or more complicated, and that's already closer to what we are discussing today, when you want to create something similar to a tissue or an organ which has to be functioning in your body, and that's much more complicated system, which have not just different types of cells, but those cells has to be organized in a special way, in a particular special way. And most important, actually, this has to be very fine-tuned, because those gene expression in different types of cell, depending on where they are located on this tissue, may be different. And they have to, if you want to do it with a synthetic design, with synthetic promoter design, you have to learn a lot about the expression, about the organization of promoters, natural promoters, which are driving genes in those different cell types in different conditions. And that's what we are going to do today in our exercises. When we talk about tissue and organ engineering, basically printing a new tissue or organ, there are already several years ago, actually already in 2016, this review was reviewing that there are experiments building blood vessels, heart, bone, liver, skin. And there are examples, for instance, for heart, when actually these iPS cells are taken from a body, and then they have to be differentiated to different types of cells working in the heart. And actually, these, as I said at the beginning, these trajectories to differentiate these cells in different types of cells are driven by different signals. So you have to do something with those cells that they start differentiated in particular direction. So you would do something, you would put on them some cytokines, growth factors, something soluble, or maybe even some metabolites which would driven those cells to one direction. Another thing which is actually becoming now very important is actually make a breakthrough now recently, very few years ago. You have to really put them on different material. Actually, whether those materials are soft or hard makes a difference, and those cells will differentiate different way. There are not so many yet, involvement of artificial promoters on artificial constructs in such a design. And I would put here additional dimension, which is not yet explored intensively towards actually making special promoter constructs. Of course, that's also related to other issues with any construct has a certain security issue. It can be expressed in the wrong place. So that's of course is all understanding of that, but still we can think about a new direction in that whole area. And now a very important area, and many of you are interested in that, is a cancer therapy, and that's something we are going to talk most today. And in the cancer therapy, this promoter design is very important, because the whole idea is let's create a promoter which would drive a gene or several genes and let's make such a promoter which would express in the cancer cells only, maybe in a particular condition. And one important condition which people found, which is actually related to cancer therapy very much, is hypoxia. It is known that many cancer cells, by growing, they increasingly become in hypoxia conditions. It's not enough oxygen for the tumor cells. And under those conditions, tumor cells appear to become resistant to therapy. They actually become even worse than in the normoxia conditions. And so on one side. On another side, this hypoxia condition is kind of hallmark of the tumor often. And there is one important transcription factor called HIF-1 alpha, which is very specific for hypoxia condition. It's actually here evolutionarily was created in evolution to switch on cassettes of genes in conditions when cells or tissues or the whole body or organ has not enough oxygen. And then in such conditions, these cells or these genes are starting expressing, and then the cells becoming, they become normal. So they find the way out. And why not, reusing this mechanism for driving artificial promoter genes? And that one example is quite recent, was published in 2023. It's very complex structure. When actually, you probably know this CAR T therapy for tumor, when there are special T cells which are artificially designed on one side, they have to recognize the tumor cells and kill them. The problem is that they sometimes actually are not able to recognize well tumor cells, and especially though recognize those tumors which have as a hallmark the hypoxia, right? And then what they did, they actually introduced in those CAR T cells specially designed promoters which are targeted by this HIF-1 alpha transcription factor, and they switching on those genes which are making these CAR T cells active. So which means they become active only in hypoxia conditions when the tumor cells are really active there, right? And then in the normal cells, they are not attacking normal cells, and that's actually the problem that CAR T therapy may attack also normal cells and have a lot of side effects and so forth. And to avoid that, so additional circuits were added to switch them on only in hypoxia conditions. That's a very interesting example published recently. Another example, actually a very old paper, it's in 2001, it's more than 20 years ago. They actually already developed a special construct, a promoter which has 6 HIF-1 alpha element, one after another, such a cassette of elements. And they showed that this designed promoter is actually really switching on in hypoxia condition in hypoxic cells. All right, so when having such a promoter, then if in a tumor cancer therapy, the main idea is to switch on so-called suicidal genes, or they call it even suicide gene therapy. So basically, under this promoter, they put a special gene which actually kills the cell. And one type of this is actually here is so-called HSV. It's from hepatitis C, I think virus, or yeah, from some virus. I have to check. And this protein actually kills the cell, and if it's expressed. And of course, you have to be careful not to express it everywhere, otherwise it will kill all cells. So that's why you need a very specific promoter, which is expressed only in the hypoxic tumor cells. And they made this experiment, and they showed that indeed in the hypoxia condition, their actually cassette, their promoter, which they designed, and they use actually similar construct like here, very simple, multiple elements of the HIF-1 alpha binding motifs, which were put in the front of the cells, in front of these genes, and they were finally killing the cells. But as you can see, it's very important to make this very specific. So other cells could be also under hypoxia, right? So, and you don't want to kill other cells which are not cancer. You want to kill cancer cells under hypoxia conditions. So you must put several signals in that artificial promoter, which will drive it only in a particular situation. So that's appeared to be a very complex problem. And I would like to actually touch here a little bit the topic about using artificial intelligence, AI, to generate promoters and enhancers. What I said in the beginning, so if we think in many, many years, we will start creating artificial life, intelligent design, but probably it will be artificial intelligent design. So it will be done not by us, but by AI. It's a little bit worrying, right? But okay. So, and it appeared to be that AI is actually a very, rapidly growing area now. And of course, it's also used here to generate promoters and enhancers. And already very, recent, or these papers, maybe already 10 years ago, these authors realized that, in fact, to design promoters using AI, you need not just a simple AI, you need the so-called generative models, generative AI. So the AI which not just simply recognize something or, discriminate, cats from dogs, something like that, but it has to generate a cat or dog. So it has to generate new sequences, generate new promoters. And then they built several designs of those AI algorithms, how to generate new sequences, which are targeted to different functions. So this is a nice paper describing different types of approaches. And generally, you have first to encode your sequence into some kind of numbers. They call it Z-latent variable. So they cannot work with letters, those AIs, they need numbers. And of course, the first idea was, okay, we have this nucleotide ATCG, 4 nucleotides, and so you can transform them into 4 numbers in some way, thinking that actually nucleotides are the letters of our regulatory code, which is wrong. Actually, I will tell the beginning. So the alphabet of the regulatory code is not the nucleotide letters. It's different. Okay, anyway, but still those approaches were quite good. So first you have to basically generate those sequences using this generative AI, and then you have to discriminate them from real, and then also train this system to find a good predictor, and then actually making this in iterations, you continuously improve your potential, your possibility to generate new promoters. First of all, you have to recognize the cat before you have to generate it. So there are a number of good latest development in the deep learning in this AI to actually be able to recognize enhancers in general. And the very good collection of enhancers is in Phantom database, and that's usually used for training to train those systems. And as you can see, for instance, this program, which is one program which was developed before called DEEP, and it was achieving only 0.82, accuracy, sorry. And then, but this new approach, which is based actually on pre-training, retraining strategies, or all kind of AI, new type of AI strategies, is able actually to increase this accuracy to 0.94, so which is a big development. All right, so but very recently, basically this year, or maybe just in 2023, like few months ago, a new breakthrough appeared at the program called BERT. So a team from US, they applied actually the big language models. So those models which you probably already know from ChatGPT, when the models of English language, or not only English, other type of languages. So language of which is written by the letters of English alphabet or any other type of alphabet. So it must be strings of letters. So and these approaches can be applied also to DNA. DNA is also kind of a nucleotide, composition. It's a long string of 4 nucleotides, and it appeared to be really, really good. So they were compared to other types of algorithms, also to ChIP-seq, ChIP-seq set. In fact, they show a bit smaller accuracy. Each paper shows different accuracy. But anyway, in this line, you can see this BERT is actually achieving the highest accuracy of predicting enhancers from Phantom database. So what are enhancers in Phantom database? These are a region of DNA in genome, which are driving expression of genes and actually expressed also by themselves. That's a Phantom type of enhancers, which are actually functioning only in specific cell types, in specific tissues, and there's a big collection of those tissues in Phantom. And when you predict those enhancers, Phantom enhancers, you also you just predict the areas where they are located. And in fact, you're a bit far yet from real prediction, but because at the end you want to predict not only the place, but also what kind of conditions this place should drive, right? Okay, anyway, so that was the biggest achievement. It was so good that even recently we got like a bigger news in newspapers in Germany. Similar project, called Grover. In fact, but on the same idea, similar idea, I would say, achieved very good, results in recognizing enhancers, and they call it even AI model to decode unknown parts of human DNA. So, which means unknown parts are actually regulatory parts. As I showed in the very beginning, those parts which encode proteins, we know, but all the rest, 99%, well, 95% of genome is unknown. Okay, but already here you see that when these algorithms start learning, and in fact, inside the bird, this already was put as a cornerstone of that algorithm, that it's not only based on a single nucleotides, it's based on oligos, on they call it tokens, on sequences, short sequences. So these is actually the alphabet of the regulatory code, not individual nucleotides, but those sequences, those motifs. Anyway, so you see this algorithm starting already recognizing importance of those motifs by themselves. Of course, but then the harder problem appeared when you don't need just to recognize the place in DNA, but you want to create a new structure which would be acting in a particular condition. For instance, you want to create recognized promoters and also create synthetic promoters, which will be B cell specific. So which means will be driving expression of genes in B cells only. And they were lucky, actually. They had, in between, they predicted generated promoters in the computer, and they had a lab nearby which synthesized those promoters, and they were learning from the experiments, so which promoters are really specific and which not, from the synthetic promoters. And they were learning it many times. At the end, they were able to actually build a model which you see here on the screen, which shows the correlation of the promoter strengths, which means how well this promoter is expressing the target gene, in the B cell, in your predictions compared to the experiment. You see, it's really correlating, and it's good, but you still see it's a bit deviations around. So it's a quite hard problem, in fact. And if you put it for the real promoters, not just synthetic promoters, you see, well, also good correlation, but still you see the problem. It's still not, even with such a good technology, which you can do iteratively and learn a lot from synthesis, and then, relearning again, it's very difficult to achieve a good, prediction. And now let me explain to you what I think about this whole area of synthetic promoters. So what we have in our brain as a kind of model of a promoter is coming from textbooks which we learned in the school, and we know that promoter has a data box, it has maybe other boxes, it has few boxes around, and then these are fixed boxes which are, and then you see transcription factors binding to those boxes. They drive the gene. And then we can think that we can apply here some sort of AI, like we have in our mobile phone, which recognizes our face. But the same way, maybe we can apply AI here to recognize the face of the promoter. So we see these different boxes, data box, CAT box, other boxes, they have a particular face. And then, right, the AI can learn from those examples and then learn the face and then be able to recognize the face. Why not? Okay, so, but let's take just one promoter. This is promoter of thin gene. It's very important kinase, which is important for breast cancer progression. And look at this promoter from a point of view of motifs, which we have in TRANSFAC. So what I showed today, so we have a sequence, and then we can have this collection of motifs in TRANSFAC of known motif or known transcription factors. We can predict potential binding sites for those transcription factors in that sequence. So you see, we can predict actually quite many places where we can find those motifs. They going one after another or even sometimes overlap. But the problem is it's like that. So it's not just one overlap. It's a mass of overlaps. So you see all kind of motifs can be recognized just in this little sequence. Just because we have so many motifs, like 10,000 motifs for almost 2,000 transcription factors, and then those motifs are all different, but they are very short. You remember these motifs had just 3C and 3A. Of course, you always can find those 3C, 3As in any sequence, and you find those many motifs. Maybe you can say that this is all just noise. You recognize something computationally and that's just noise. Okay, but if you look at the real data, so you know this ChIP-seq experiments, it's not prediction, it's real facts. So when you take transcription factor and you actually cross-link it to DNA, and then into that place where it was sitting in a particular cell type, and then you sequence these fragments of DNA which were actually target for that transcription factor. So we have a database called GTRD, and there are a number of other databases where, and also TRANSFAC, we also collect ChIP-seq data. And if you just put those known ChIP-seq peak for different experiments on the same area, you see the same picture. So it's not the artifact of predictions. It's a real fact. So these transcription factors in different conditions, indeed binding to this place in the same place. Like they don't have any other places. They all want to sit on that transcription factor binding sites in this promoter. This promoter is very important for them to sit, right? But let me ask you, and I already will give you an answer. So what is a common between all these people on these pictures? And you can tell me that probably you can recognize that all the faces are different, but all of them are actually smiling, right? So there is one feature, not the whole face, but just one feature of that face, which is similar, the smile, right? And maybe the same here. So we have all this potential in this gene to be bound by different factors, but in one particular condition, only some subset of those motifs actually acting together. It's like those muscles in our face which create a smile, right? But how to recognize that? And in fact, that's a very difficult AI problem. So there are some tools which can recognize those emotions on the face. This is my face, the same face, like here. But you have different, like here I'm smiling, here I'm curious, here I'm like sad or I'm surprised. And this tool has to recognize here the smile, it recognizes pretty well. The curiosity, it's actually recognized wrongly. It thinks I'm actually kind of in a bad mood. So sometimes it's so it's not easy to recognize the emotion. So that's why we need an algorithm which would recognize not the whole face, but recognize an emotion in that promoter. So promoter emotion actually. And the promoter can have different emotion, can smile, can cry, can do different things, and can do millions of different emotions in different conditions. And different parts of DNA, different motifs will be involved in those different emotions. And some years ago, we created this algorithm, which is based on genetic algorithms. It's some kind of pre-ancestor of AI. What it's doing, it's simply scanning those promoter areas with all these thousands of motifs, and it tries to find an area, some window, some small window, which has a particular composition of motifs, which we actually don't know. It has to find that particular composition in the sliding window. So, and this algorithm is working quite well. Actually, you put there a set of genes, a set of promoters, which have the same behavior, like they all smiling or they all grinning in the cancer, we'd say. And then this algorithm will find those motifs which are common between those promoters. And to do this automatically, we have actually a tool called Genome Enhancer, where you can put just drag and drop transcriptomics data. So here we took a data from cell line under hypoxia conditions, because we know what is a HIF-1 alpha binding site, and of course we can repeat those sites many times, and this will be artificial construct, artificial promoter, but it will be always working as soon as HIF-1 alpha is activated. So in all kind of hypoxia, even in other type of cells, not only in cancer cells. But we want to have such a construct which would work just in the cancer cells under hypoxia conditions. So to learn this type of construct, we have to take this condition. So we took this cell line. This is a cancer cell line. It's glioblastoma, actually. Sorry, I already jumped a bit further. And we took this transcriptomics data, put it in the Genome Enhancer. It analyzes it automatically. And actually, it builds this model which discriminate promoters of upregulated genes in this glioblastoma cell line under hypoxia conditions from the other genes, which are actually not changing the expression. And you see the top one is so-called VEGF-A, which has those number of sites in a clustered manner. And this is a model which is system found. And you see it found actually HIF-1 alpha motif, but it's accompanied with by other motifs for the other factors. So like, SMAD3 and SERBP and AP2 and other factors. So, and then only in that composition, it is specific for that particular conditions. And this system is also building the whole, network, showing how this VEGF-A through HIF-1 alpha activate the whole system, and based on this, feedback loops and so forth. So we were discussing that some in previous coffee breaks, how it's actually building such a network. But for today, for us, what is important is basically we built a system which can recognize such an event. So we know that indeed in the cancer cell under hypoxia, this HIF-1 alpha is activated, and it actually has the binding sites in which is already known in VEGF gene. And the VEGF gene is then produce the protein which is activating other cells through the receptor, and those other cells give a signal to cancer cells to grow. So it's like a feedback system, and then the system is the cancer cells start growing more rapidly or becoming more resistant. So we actually identify this this kind of mechanism just by analyzing transcriptomic data. But for what is important for us, by doing that, we actually learned how the promoters of those VEGF genes could be organized. And for instance, we found that this HIF-1 alpha is accompanied by very often but SMAD3 factor binding sites, which are located nearby and sometimes even a little bit overlapping. Well, in this case, they are really, face to face. We actually, there are a number of articles saying that indeed those, the HIF1 alpha factor is cooperating with SMAD3 in many other genes. All right, but for the exercise of designing new promoters, we will take another data from another factor, which is called AML1. I was talking about that today in the beginning, or RANX1, which is actually important for many different, behavior of bad behavior of cancer. For instance, what is important is it's involved in metastasis. And one of the goals of cancer therapy would be, for instance, to create such a promoter, which would be, acting only in the cancer cells, and only in these conditions when those cancer cells become metastatic. So because metastasis in cancer is a really problem. All right, so in this case, why I took this data, because it's not only gene expression data, but it's also ChIP-seq data. So these are two types of data which can be combined together, and that gives even more power to predict and more power to design new promoters. So to make this long story short, just we found out that this gene, which is called EYA2, it's very rapidly upregulated in case if basically it's actually downregulated if we knock out AML1 transcription factor. So AML1 is upregulating this gene in the metastatic cells. So then we analyze that all these genes which are acting in metastasis and build this promoter model which appear to be having this AML and RANX2 motifs inside, but also other factors like glucocorticoid receptor binding sites, STAT sites, and then when we start looking at this gene in the chromosome, we now see it's not only promoter, but it actually has an enhancers inside the intron. If you look inside these enhancers, so for instance, this enhancer number 3 in the first exon, it's actually in the third intron of that gene, it contains those motifs for AML1 transcription factor and accompanied motif for other factors. So more in zoom, you can see that this AML1 is actually duplicated in this area, and glucocorticoid receptor binding sites in nearby. And we have here also STAT1 site overlapping with GR site. And this construct is a kind of a core of that enhancer. So why not actually taking this enhancer and try to build a new sequence and try to work with this enhancer a little bit to create a construct which would, have this property of be active in metastatic cells. So for that, we actually now will go directly into our tool. I hope you see now my screen of the Genome Enhancer. In fact, I already looked inside this Genome Enhancer in the body of the tool, so where I don't see just the final result, but I can look inside, what kind of intermediate result it has. And indeed, here, as I showed to you, we have this. The system created this this model for the enhancer working in metastasis, and you see this, it has this RANX and AML motif, as I said, and it discriminate those enhancers from other areas, from the non-active areas of genome. Right, so but now how to use this model to generate new promoter? So what we did actually, we took this promoter of this one gene, this EYA2 gene, and we put it in as a FASTA file. So I will show you in a moment. So we took this enhancer from that gene, we put it as a FASTA file. And then what we did, we start, playing with this FASTA file. So for instance, I took this AML1 binding sites and just repeated one time. Another thing what I did actually, I make it shorter. I removed the spacers between those places. So for instance, if you look here, so if you have a gene, and you can see these more details, you see that sometimes the sites are quite far from the center of that enhancer. So if you remove a part of the sequence and make it closer, maybe it will make enhancer better. Who knows? So that's why I'm starting to kind of play with this sequence by duplicating those sites. Then I also duplicated glucocorticoid receptor site. And then what I did also, I actually changed glucocorticoid receptor sites to make it better. So it is natural site here, but now I can put certain mutations in it to make it closer to the consensus of GR binding sites. And then I can put these sequences inside the platform, and here they are. So here we have these sequences. So for instance, this is a promoter. This is a, with a sequence with duplicated AML1 and glucocorticoid receptor and all other kind of variants. We have all these variants, and then we can look basically how these variants are, behaving from the point of view of that motif, of that model. So, and the measure of the model is actually the score. So you see, we have this score. So if you look now at the score of the real promoters, so which we had a look just before. So we have here it is. So it's here. So we have this score. This score, and you see the very good enhancers. They have score around 15, 16. And actually the natural score of that enhancer we're now looking at, enhancer of that gene, is around 11. It's 11.6. It's somewhere here. So it's still far away from those places of genome which have no, have nothing to do with such type of enhancer, but it's not the maximum, right? So it's around 11. So now after changing these nucleotides in that sequence, I can look basically whether my changes improved the score or made it worse. Okay, so let's have a look. And indeed, now what I see, the score of, we get actually higher score, 15.19, for a variant where when I actually duplicated AML1 site and GR site. So I just simply duplicated them. But I was interested to look at that variant which had a glucocorticoid receptor modified. I made it actually better, but in fact it decreases the score. And I was interested why. And the reason was, in fact, because if you remember, these glucocorticoid receptors overlapping with other important sites, and when I changed the nucleotides in the GR, making this consensus better, I actually made consensus of other sites worse. And in the balance, it appeared to be less favorite for this model. So, but anyway, so this way you can see how this can be done. And of course, this could be done also automatically. For instance, we also implemented a program which generates simply sequences using a very simple algorithm. It actually takes an enhancer of a gene and then try to create a new sequence simply using the same composition of words of the length 8. And then it created to us this set of sequences. They are here. They are. And then I applied the same algorithm here. See, these are purely artificial sequences, have nothing to do with real promoters. And you see even just a random generation with such a little bit a smart way already give a score which is a closer, very close to the one we have in the good enhancers. And by improving a little bit of that, composition or removing some of the, of the spacers, we can, in fact, even increase this score further. So, and we have several other tools to manipulate with sequences to create new enhancers in such a way. But still, this is so far the kind of intellectual, intelligent design by our intellect. We would like to apply also artificial intellect to do more of all those designs, and that's a little bit of our future. But now to finish with this part and with the talk today, I want to show you the two final slides, and whether we actually a bit closer to understand what is a regulatory code. Maybe we can, in fact, read this message, which is there, by taking into account what we learned so far. So, but actually what we learned so far is something like that. It's time now, the autumn is around, the cold temperature is coming, and you often see on our, window such a picture of the condensate. When the water is condensated in the glass in such a nice way. And it appeared to be in recent research that the same way the transcription factors are actually binding to DNA, like these droplets of water binding to the surface of our window. The same way, actually, those transcription factors, based on this phase separating a principle, they form those droplets, those condensates, together with DNA in the nucleus. And you can imagine how difficult it is to predict where those droplets will form on the glass. You know how variable it is. You put a little scratch here, and you get more droplets there, or you put here and there. So it's very flexible. That's why to predict the promoters, to design new promoters, is such a hard work. But basically, what we have in mind is something like a fuzzy puzzle. You actually have a surface of DNA like a glass, and those proteins, by soft touch, they don't have to have a long motifs to bind. They have a very short motifs, so they have a very soft touch to DNA. They can organize those droplets. And if there is something happening with DNA, for example, a mutation or just epigenetic changes, it can change the environment of that glass, of that DNA, and other droplets, other transcription factors are accumulating there. So that's a very flexible system. And taking this in mind, we can now read this message, which was here, like in very bizarre language. Basically, what we have to do, we have to read it by closing the part we don't understand, and read only those letters we understand. So, like we were defining only smile from the whole complexity of the face. So we can read, for instance, hello, Alexander, or we can read in the same message, completely different message, goodbye, Alexander, or run, Alexander, fast, and other messages. We have tons of different messages in that DNA. And the more random, actually, the sequence is, the more random, the more closer the promoter to the random sequence, the more messages you can encode there, right? So, and that's the whole, idea of that complex system, and our big challenge to understand that. All right, so you see today was very little of demonstration, but a lot of words from my side. So the first question, I have developed a novel algorithm for genomic classification problem, which take chimeras and provide near 95-100% accuracy. It can be applied in all area, even junk DNA, and I have confirmed it. Very well. Wow. Thank you for, I believe this is a key to work with chimeras. As you can see, there's algorithms like DNA BERT. As soon they start working with words, not just with nucleotides, but with words, they achieve this much better accuracy. And as you are writing here, you find a way how to work with chimeras. Very good. So if you have this algorithm, I would be very interested to look at it. You gave me the email, so you can write also to me directly. I will write to you after this talk, and yeah, let's keep in touch. If it's making so good, so let's try to apply it to real problems. Let's create a promoter which will kill cancer, right? Let's do it together. Thanks a lot. I will copy your email. All right. So then, actually, I have a couple of other questions. So, for instance, I got a question, a very philosophical question, I would say. We can now create artificial blood vessels, skins, and even noses, right? But what are challenges remain in growing more complex organs like liver or heart? How much more difficult it is, and what steps must be taken to achieve this breakthrough? That's right. That's a very interesting, actually, topic. So to answer this question, yes, bigger organs like liver and heart, and you already seen that people are starting to think and actually making attempts to develop something like heart, right? But of course, there are a number of problems. For instance, so many different cell types. And sometimes we don't even know what types of cells are there. So because there may be a very little subtypes of a cell type. To know all the cell types and just take, for instance, immune cells, which are inside the heart, anyway, and they also like thousands of different types of immune cells. We have to, in fact, learn first what kind of cell types are there. But even knowing what cell types are there, another complex thing is actually these cell types, they changing. So while they are in the organ, when they are functioning, the organ is functioning, these cell types actually have to be able to change over the time, depending on an organ. So in the heart, maybe it's more mechanical, but for instance, the liver, it's more longer processes, and those cells has to function. It's not just it has not just the form. Those cells has to function, has to respond to in the right way to toxins and everything what liver is doing. And of course, to be able to grow such cells from iPS, which have not only just forming a typical type of the cell of our target, but also behave in the right way. That's getting more complicated. So maybe in that way, in that problem, these artificial constructs could be very useful. And of course, 3D structure, 3D architecture of organ is important. We know all that nowadays it's becoming a very important topic. This spatial transcriptomics. So the same cell type, depending which place it is in the organ, it can switch on and off different types of different types of genes. And also to take into account this space is a complex thing, although we know that this 3D printing may be a way toward that. But actually, in fact, we can list all the problems at least. And since we know the problems, probably we'll find a way how to solve them. And then, maybe not, we shouldn't wait so long until this will become possible. So at least I don't see any kind of hurdle in between, so that will stop. Definitely will stop the development, and we don't know what to do. So actually, we see the direction, we know what to do, and we have to just do it. All right. So there was another question. Let me see. While designing artificial promoters to impact the gene expression, are SNPs in the core promoter or upstream regulatory regions considered as a potential target and given priority, or only the modification of a given sequence in these regions taken into account to create such promoters? All right. That's a very good question. Actually, that's a very important question, and it triggers to me to a new type of development. Indeed, so far our algorithms, when we design, the algorithm, for instance, to find the regulatory module which drive the gene expression, like in this case, we take reference genome, right? So we take the promoter or enhancer areas of the reference genome, and we predict the the score of enhancer, like this one, for the sequence from the reference genome. But of course, there are like millions of SNPs, and we people or other organisms, they carry different genomes, and they're all okay. So many SNPs which we observe, many variations in genome, they're kind of neutral. Majority of them, I would say. And there is a kind of rule of thumb. So if you take SNPs whose frequency are above 1% in the human population, we would postulate those SNPs are probably neutral, which means if you have a change of nucleotide in that position, the enhancer should still work, right? So which gives us a lot of potential to increase the set of genes, set of enhancers we have to learn our models on. So now we are learning our model maybe on 300 enhancers, but if we add all these SNPs, we can increase it to thousands and millions and learn. But of course, we have to take into account in this learning algorithm the bias, because the same sequence is many times in that set of training set. But that's possible to take into account. Yeah, but that's a very good idea, and thanks a lot for this idea. We'll take it into account. All right. So there are so far all questions which I had. This is just a start. You see, today we had just a lecture, but I want to show you more tools in our portfolio in that direction. The direction is, like artificial promoters, like in general, how to design sequences which would do what you like. Let's create life, Keep in touch and see you next time.